Hey everybody. How's it going, Lillian? Are you home still?
Yep, I'm
You didn't leave?
nope.
No? Is it gone? Is it over now or
No, it's actually just starting.
just starting? Yeah, that's what I thought. Well, good luck.
Thursday's gonna be when it's at its worst.
Yeah.
But we're at 73 feet above the sea level.
Oh,
So
okay.
if we flood, there's a big the whole state has a humongous problem. Um,
That's true.
but we and most of Florida's underwater. We shelter in place so that the coastal people can evacuate because they
Yeah,
have to evacuate. So we
they
shelter in
do.
place so that we don't get interfere with their ability to get out. We're the
Yeah,
place that people evacuate to. Yeah.
right. High ground, exactly.
Water
Exactly.
plant.
Well, good luck.
Although
I hope
it's
you don't
not
lose
very
power
healthy.
or anything. Yeah. All right.
It's
Hopefully you
a
guys all
suffering
got
water plant.
my what, Lillian?
Suffering water plant.
Yeah. Yeah. Hopefully you all got the message this morning. Another good example of why you need to check your messages before we have our live classes. So that's always important to do because I will put any information that you need for class there. As I did this time, if ever there's anything that you're not likely to have around your house, I'll give you more warning than just the morning. But like today, you needed to bring a couple more things than I said last week. So the foil and the marker and those kind of things. So, all right. Anybody have any questions before we get started? All right, let's dig right in. Yeah, high ground. All right. Here we go. I don't know why, because I'm not in the path of the hurricane, but I've had a lot of internet trouble today. So I'm just giving you guys a warning. If by chance you lose me, give me a minute. I'll be hooking up to my laptop and trying to get onto use my phone as a hotspot, but that takes a few minutes. So always give a teacher 15 minutes to be late or to reconnect if there's a problem. If I'm not able to make it back in 15 minutes, that's okay. It's been okay for my other classes today, but this morning was all kinds of trouble. So, all right. So hopefully you can all see that. Honors biology. Biology is life. Of course it is. All right. So we're going to talk a little bit about that egg osmosis lab that you did. I thought this was a cute picture. Wouldn't it be nice if we could learn that way? You just lay your face down on a book and and you would know it. Unfortunately, it doesn't work that way. All right, so let's talk about eggs because eggs, or rather, an egg is a cell. Eggs are the largest cell that most organisms make. And that's because egg cells contain all the organelles, the energy sources, and all the cell parts that are needed for the egg to grow into an adult. Like everything is in the egg. The only thing that the sperm contributes in fertilization is DNA. Everything else comes from the egg. So the egg has to be really big. Now, bird eggs are some of the largest single cells on Earth. Once they're fertilized, they start growing really fast. They will divide very quickly into billions of cells. And those billions of cells, of course, make up the tissues, and then the tissues make up the organs, and the organs make up the organ systems, and then the organ systems make up the organ finally before the organ system of the organism before it finally hatches. So we use chicken eggs in this lab in order to give us a model so that we could see how a cell is affected by the things in its environment. So the very first thing that we did with our eggs is we put it in a solution of vinegar and we let it sit for a few days. So why did we do that? What did the vinegar do? Shmuel.
It melted off the outer shell, which
Okay, good. Don't
dissolved.
like the use of the word melted though. Find another word.
Dissolved.
Yeah, there you go. There you go. You're absolutely right. Just melted refers to heat. So raising the kinetic energy, the thermal energy of something, so that we're not going to use that term. But you're right. It gets rid of the same way. So dissolve, we say it gets rid of that shell. So chicken eggs, like most eggs, are made like most hard-shelled eggs, are made of something called calcium carbonate. It's actually the same material that's found in seashells, and even marble is made out of this calcium carbonate. So once that shell was gone, what happened to the egg? How did the egg change after the shell was gone? Go ahead, Lillian. I saw you making the hand motions.
Yeah, it got sort of like goopy and
So
just
right immediately after the shell
not very
gone,
rigid. It got the opposite of rigid.
in the vinegar still?
Yeah, it was already sort of like goopy
Huh, that's interesting. Usually they get
because
pretty
the shell
pretty
wasn't
firm.
holding it. Interesting.
Yeah, yeah. We did have a couple of different effects. Did anybody's egg get big? They got larger, and they were kind of, at least, most of the time, it's kind of feels a little bit like a water balloon, like a filled up water balloon. So it's got that little bit of pressure that's pushing back. Why? Why did it get bigger? Once the shell was gone, what happened to make the egg enlarge? Something had to happen. Shmuel,
There was more water in the vinegar than in the egg.
I'm really barely hearing you. Can you speak up or turn up your?
There was more water in the vinegar than in the egg, so there was diffusion and water went into the egg.
Okay, I think I heard what you said. It's kind of hard to hear though.
Yeah,
There you go. That's it.
yeah. I changed the volume for the computer.
Okay. Okay.
There was a higher concentration of water in the vinegar, so by osmosis, more water went into the egg than went out of it.
Okay, you're almost exactly correct. So it really osmosis has less to do with the concentration of water and more to do with the concentration of particles. So you're right that there was more water in the vinegar than there was in the egg, but there was more particles inside the egg than there was in the vinegar. So what water tries to do in osmosis is, you can think of water as being the social distancing police of the particles. It wants to get them to spread apart. It wants them to be the same on both sides, not the number of particles, but the concentration of them. So water moves in order to cause those particles to spread apart. When the water moves in through osmosis, we get a bigger egg. It spreads it out. And because the membrane is flexible, it was able to take that movement. All right, so then the next thing you guys did was pour out the vinegar and you covered the egg with Karo syrup. What happened then? What happened after it sat for a day or two in that Karo syrup? I know you guys did the experiment because I saw your pictures. All right, Yaya, are you wanting to answer? Get your hand up. I can't tell if it's a clap or your hand.
It deflated.
Yeah, it did. It lost water. So once again, be careful with the words. Deflated generally refers to air, and air didn't leave it, but it definitely got that deflated look about it. It got smaller. It shrank. So what happened was osmosis still happened, but it happened in the opposite direction. So think about the corn syrup, that Karo syrup that you used. It's mostly sugar, and it has a lower concentration of water than the egg does. Now, to reach equilibrium, the water moved out of the egg to try to spread out those sugar particles. So once again, the water went, I got to spread these guys out. I have to social distance them. We all hate that word, don't we? But it works for osmosis. It works because it moves them apart from one another. So it moved out, the water moved out of the egg until both solutions had the same concentration of water. Now that outward movement of water is what caused that egg to shrivel up. Shmuel.
Well, I noticed also that the Karo syrup got more watery, especially where the egg was, but also in the control with only the water. I noticed that after like a certain point, it wouldn't get any bigger.
Right.
Like it
Yep.
was.
Absolutely. After a certain point, it's not going to get any bigger at all. So you're right. The reason that Karo syrup got a little more watery is because water was going into it from the egg. Water was spreading those particles of sugar apart. Now, there were a couple of you guys who wrote to me to tell me that your shell didn't seem to be dissolving and you weren't really sure what to do. Anybody who wrote to me from here and said, my egg isn't working? All right, maybe it's nobody who was here in this class now. So the interesting thing is all of the non-dissolving shells were brown eggs. Interesting. So, the scientist in me went, ooh, that's a good a good question that could lead to some scientific experimentation. So I decided to run an experiment of my own to see if there was a difference in the way the brown eggs and the white eggs behaved in this experiment. So I'm going to show you what I did here. This is what my eggs looked like at the very beginning, before they were placed in the vinegar. I had one white one and one brown one, and look about the same. They're about the same size. I specifically chose ones that were approximately the same size. And you can see how much of the spoon underneath is showing in them. And I have a yardstick there so that you can see about the size of them. Any of you guys know why some eggs are brown and some are white? What makes a brown egg? Pretty simple, actually. It's a brown chicken. So different breeds, it's due to the breed. So white eggs come from breeds that have white feathers, and brown eggs come from breeds that have brown feathers. Simple as that. All right, so I took these eggs and I put them down in some water. And this is what I started to see right away. I started to see those bubbles forming. What do you think the bubbles are made of? Air, kind of, yeah. There's something in the air. There's specific things that they're made of. Same thing you breathe out.
Carbon dioxide.
Yeah, carbon dioxide. Yeah. So vinegar dissolves away those eggshells because it has a thing in it called acetic acid. And that acetic acid reacts. It makes a chemical reaction with the calcium carbonate that's in the shell. So that reaction, so we start with carbon dioxide and acetic acid and calcium carbonate. And we, in this reaction, take apart the molecules and rearrange them. What we rearrange them to make is something called calcium acetate, carbon dioxide gas, and water. So we're seeing the bubbles of the carbon dioxide gas rising to the top. You can see that reaction happening. All right, so after they had sat for 2 days in the water, that's all it took for mine, they looked like this. Now you can kind of see why people who had brown eggs thought that it didn't dissolve. Doesn't look like the brown shell is gone, but look how much bigger it is. Remember before we could see the spoon under the sides of it, and they're both significantly bigger. So this shell was dissolved away. Both shells were dissolved by the vinegar, but the membrane itself is brown in a brown egg. So it didn't become translucent like the white egg did. But since the shell was gone, it did allow water to enter the egg through osmosis. All right, now we're going to compare the eggs here. This is before and this is after. So you can see the size difference. They both lost their shells. They both got a little bit bigger. They both were soft and kind of resisted the pressure that I applied with my fingers. Now, there was no difference between the two eggs other than the fact that the shell was brown and one was white, but they look a lot different. The brown one still looks like it has a shell. So now after this, I poured out the vinegar, I covered them with Karo syrup, and I left them for 24 hours. This is what happened after 24 hours. Somebody make an observation for me. What do you see? What do you observe here? Shmuel.
That when you first had the eggs, that the white one looked like a tiny bit bigger, and then when you took them out of the vinegar, the brown one was bigger, and after you took them out of the Karo syrup, the brown one was still bigger.
Yeah. Yeah. So what does that tell us? What happened? What happened to the white egg that didn't happen to the brown one? Yeah, the brown egg didn't shrink. Why not? What causes the shrinking? Oh, you're on the right track, Luke. But remember what we call diffusion of water through a membrane. Right. Osmosis is what happened. Exactly. Osmosis is diffusion. So you're right about that. But it's so important. The diffusion of water is so important. It gets its own name, osmosis. All right. So here you can see the way the white one really shrank, while the brown one is soft, but it didn't lose as much water as the white one. It didn't release. Look at how soft that one is. Really lost a lot of water. Yeah, you're right, Shmuel. Almost as if it lets water in, but not out. Any ideas why? What would you look for if you were trying to figure this problem out? Just take a guess. Because the truth is, I didn't know either. I had to take some guesses and then do some recent researching of things. Great, the type of the membrane. What is the brown pigment made of? Yep, the membrane. Yeah, so that's what I thought. So I read some studies about this to see if I could find a difference between the membrane of a white egg and the membrane of a brown egg. And here's what I found. So we started with 2 eggs. This experiment that I read about started with a lot more than 2, but let's say they had some brown eggs and some white eggs. They were all the same size and they compared them. Eggs that were brown weighed more. Exact same size, but the brown eggs weighed more. Why do you think that might be? Denser. Yeah, exactly. They're actually thicker. So they're thicker and denser than a white egg. If you've ever cracked a brown egg and a white egg, you can tell it takes a little more force to crack a brown egg than it does a white egg. Another thing was that the brown eggs had what's called greater static compression shell strength. What do you think that means? Static compression shell strength. How would you test for static compression shell strength? Eleni?
Dropping them or trying to crack them with the
So
same
you're
force.
dropping them, probably not, but trying to crack them with some force. How much force can I apply to it before it cracks? Yeah, we want to have them want to see which one is more resistant to pressure. So this is what they do. This is how they test for static compression shell strength. They're putting this pressure on it and seeing how much pressure they can put on before it breaks. So the static compression shell strength, that talks about the ability of that shell to withstand that force of compression without breaking. Now, shell structures are real thin, but they're curved. And curved surfaces are really good at distributing the load. So when you push down on this part right here, the load is spread out all the way along the curved part right here. Where something that's flat, it's not going to be spread out like that. So they use curved surfaces in a lot of engineering design. Buildings, airplanes are all curved, and this is part of the reason why. So a lot of things that have a curve are just simply stronger. Soda cans, yeah, absolutely, because they've got greater compression strength. Another thing that was different about the brown egg and the white egg is the what's called vitelline membrane. So find the vitelline membrane on that. It's this one that separates the yolk from the albumin, which is the white. The albumin, the white is made of a protein called albumin. So the vitelline membrane is stronger in a brown egg than it is in a white one. So is this significant for the experiment we're doing? I got a couple yeses. Any no's? Okay, we got to know. Why do you think no? Maybe. That's diplomatic, right? Diplomatic. Actually, it's probably not all that significant in this experiment because we're concerned about this membrane right here. We're concerned about water going, we took the shell off. Now we're concerned about water passing back and forth across this membrane. There could be a little bit of diffusion of osmosis across the vitelline membrane, but honestly, there's very little. There's very little because that's what is going to grow into the chick in this case. And the white is what's going to nourish it, has more water in it. So actually there are, it seems like yes, because it's a membrane, probably because we care about, yeah, yeah. Well, you might think so, but no, there might be a small impact from this vitelline membrane, but we're really mostly concerned about these outer shell membranes. So there's an inner shell membrane and an outer shell membrane right here next to each other. And they provide a couple things to the egg. First, they're just a physical barrier between the egg and its environment. They do a lot to help to protect the egg against bacterial infection. And once that egg is fertilized, they really work to regulate the exchange of oxygen and carbon dioxide across this. Now, what's interesting is when they tested the membrane, there was no significant difference between the shell membranes of a white egg and the shell membranes of a brown egg. Okay, so now let's look at all that we just learned. We learned that the shell is thicker in a brown egg. That would certainly impact the speed at which it dissolved. But once it dissolved, once that shell was gone, since there's no difference between the composition of the shell membranes of the white or the brown egg, we would have expected them to behave the same way, but they didn't. So for some reason, there was less movement of water by osmosis out of the egg and into the Karo syrup than there was in the brown egg. So really, I didn't get an answer. The mystery continues. Now that led me to a whole bunch more questions, which a good scientific experiment would do. So here's a hypothesis to take this a little bit farther. If I changed the Karo syrup, got rid of the watery Karo syrup and started with fresh and left it for another 2 days, then I would see movement of water across the membrane into the Karo syrup. That's a possibility. That is a possibility. It's also possible that just because we don't know what's different about that membrane, that doesn't necessarily mean there isn't anything that's different about it. So we continue learning. I'll tell you what I did learn though. I will tell all future classes to use white eggs so that everybody starts with the same egg and you can see the difference. All right, anybody have any questions? All right, great. We're gonna now do, oh, go ahead, Shmuel.
But there must be some difference because one's brown and one's white. So
Right. Well, there's at least a pigment difference, and that pigment may have made a difference, but it didn't seem to make a difference in the osmosis into the egg. Remember this one right here?
what
They seem to be about the same size.
could the protein shape like make it allow stuff in but not out, like a artery or a valve on the heart?
So yeah, you have membranes, proteins. Those proteins that are embedded in the membrane that let things go in and out. And comparing it to a valve in a heart is actually a good analogy. It's something that would open up and then close up. And there are some proteins in the membrane that do that. There are also some proteins in the membrane that just are tubes that go through. They don't open and close. And some things can just move right in and out. Now, water is one. Water is a substance that has a pretty special way that it moves in and out, but water can go right through the membrane. It just can't go through fast enough, so we have special channels that sort of pull it in. They use charges, like positive and negative charges, to pull water across the membrane when it needs more water. Those, we don't know. This is where I say more research would need to be done. I don't know if anybody's researched the protein content of the shell of the membrane of the outer membrane of an egg, of chicken egg before. I don't know. That's a possibility. So I wonder what would happen if you used an, all right, so what is an ayam samani chicken egg? A different kind of breed of chicken?
The entirety of the chicken is black, even its insides and its eggs.
Wow. So eggs
Yeah.
do come in a bunch of different colors. You can find pink eggs and blue eggs and green eggs. Yeah, that's pretty cool. That's pretty cool. Yeah, excellent, excellent thoughts. All right, let's move on and do a little prep before we move into our experiment. So last week you guys learned about photosynthesis, and this week you're learning about a process that's really in a lot of ways the opposite, the reverse of photosynthesis. So hopefully you were able to get the supplies. If not, it's okay to observe, but you all need to fill out that lab sheet that I sent you this morning. So even if you're not going to do the experiment yourself, be ready to fill out the lab sheet. So when we're talking about photosynthesis, we have reactants that go in and we have products that come out. Anybody remember what the reactants are? What goes in to photosynthesis? Shmuel.
Carbon dioxide, light, and water.
Yes, excellent. So when we say product or reactants, we're usually talking about matter. We're talking about things that are actually made up of atoms and molecules. So the reactants would be carbon dioxide and water. Light is necessary, but light is not matter. So we would say in the presence of light. So that's where that light energy would come from. It's not usually written in the column along with the other reactants necessary though, especially for that light, well, only for that light dependent reaction. All right, now we start out with carbon dioxide and water. What are the products? What comes out of this process? Yeah, you got it, Eleni. Glucose and oxygen. That's what comes out. So photosynthesis is this process. Light energy from the sun is converted into chemical energy. Energy can't be created and it can't be destroyed. Same as matter. Matter can't be created, can't be destroyed. So this process converts chemical energy, the light energy, into chemical energy in the form of sugars. All right. Now, that energy that's locked in those glucose molecules is crucial for living things. That's where we get our energy, is ultimately from the process of photosynthesis. Now, you guys remember the 2 stages of photosynthesis? There's 2 stages. Anybody except Shmuel? Not that I don't want to hear from you, Shmuel, but I want you to have to do all the work. Somebody help him out. 2 stages. Oh, go ahead, Luke.
Is it the Krebs cycle?
It is not the Krebs cycle. That's coming up. That's part of cellular respiration, but well done for knowing that. That'll come in handy. Lillian?
The Calvin cycle?
The Calvin cycle. Which one is the Calvin cycle? The first one or the second one?
The non-light dependent one.
Okay, so then what's the other one called? Krebs cycle and light dependent. That's the name of it. The first one is just called the light dependent reaction or the light reaction. The second one is called the Calvin cycle, or sometimes you'll hear it called the light independent or the dark reaction. I know it's confusing when things have a lot of different terms, but there you go. All right, so take a look at that light reaction. Now, ATP stores energy right here. That stores energy, and NADPH carries electrons. Now, this whole thing needs light. This will not happen if there's no light. It also needs water. But look what it doesn't need. It takes the oxygen from the water and it just kicks it out. All it really needs is the hydrogen that comes with the water. So it gets rid of, it ejects that oxygen right at the very beginning of the light reactions. And that's a really good thing for us because we need oxygen to breathe. We're in a really wonderful symbiotic relationship with plants, with things that perform photosynthesis. We need oxygen, they give it to us. They need carbon dioxide, we give it to them. All right, now, just the hydrogen that's in that water molecule is needed. And it goes, see this NADP? Picks up the hydrogen, now it's NADPH. All right, now the ATP and the NADPH from the light reaction move to a different part of the chloroplast. The NADPH gives up one of its electrons. Now it's just NADP+. The ATP gives up some energy. Now it's ADP. And the electron and the energy, this electron, this energy, are used in order to rearrange the atoms that came in the water and carbon dioxide and are given off as oxygen and sugar. We just got a rearranging of molecules. All right, now that we got a little bit of a review, get out your lab sheet. So when we do a lab like this, whenever you do any kind of a lab, the very first thing that you need to do is read all the instructions. Okay, so I have a little side story for you. This happened to me last night. So I was sitting at my desk here and I was trying to fill out some tax information that I have to do. And I really don't like reading government papers. They're very hard to understand. So I was reading it and trying to figure out what exactly I was supposed to do. And I was talking to my sister on the other end of the phone. My sister is a scientist at the University of Iowa. And she said to me, "Did you read the entire document from the beginning to the end?" I said, "No." And she said, "Any good scientist knows you always have to read the whole thing before you get started." So I learned a lesson yesterday. But yes, we always want to read the whole lab sheet. So let's start by reading this. What are we trying to do here? That's our objective. We're trying to observe how plants utilize carbon dioxide and release oxygen during photosynthesis using bromothymol blue, a pH indicator, to detect changes in carbon dioxide concentration. Here's our background information. This is what we need to know about this lab. Bromothymol blue is a pH indicator that changes color depending on the amount of carbon dioxide present. In neutral conditions, the solution is blue. As carbon dioxide dissolves in water, it forms carbonic acid, which lowers the pH, turning the solution yellow or green. During photosynthesis, plants take in carbon dioxide from the environment to produce oxygen and glucose, which can cause a decrease in the amount of carbon dioxide in the surrounding solution. All right, what's pH? It's all over that. What is it? What do we mean when we say pH? Lillian.
Acidity or alkalinity?
Yeah, alkalinity, but yes, you're right. Yeah, it's basically how acidic something is or how alkaline it is. And yes, Marianne, base and alkaline mean the same thing. So you can say how basic it is. That's exactly the same thing as alkaline, right? So acids have a low pH and bases have a high pH. Yeah, you got it, Shamil. Potential of hydrogen is what pH means. I'll show you what that means here. So this chart here shows us the pH of different substances. So something has a high pH. Look this way, goes up, the numbers go up when it is basic or alkaline. Bases have a high pH because they have more hydroxide ions in them. Now these hydroxide ions make the substance less acid and more basic. Something has a low pH when it's more acidic. Acids have a low pH because they have more hydrogen atoms in them. The more hydrogen atoms there are, the more acidic the substance is, so the lower the pH is. So I remember the difference. The way I remember this, I think of where hydrogen is on the periodic table. This is where hydrogen is right here. It's little. It's a little number. It's a 1. The smallest element is hydrogen. So I remember that the smallest pH numbers have more of the smallest element. They have more hydrogen. So then the highest numbers are going to have less hydrogen. They're going to have more hydroxide. Just the way that I remember those. I like to look for little clues that help me remember stuff. All right, so this is the color of bromothymol blue in water at different pHs. Right there. So that would be water and some other substance that is changing the pH. That's just for your information. We're not going to test pH this time. All right, now the next thing we need to do is look at our materials list. So here are the things that you need. Safety goggles, check. Always have to wear safety goggles. If you don't have any, I recommend getting some. We will do a couple more experiments this year where having safety goggles is important. You need to protect your eyes. Bromothymol blue is a solution that can be an eye irritant if it's splashed into your eyes. So we always want to wear safety goggles. You need the bromothymol blue, a small water plant, 3 test tubes or small jars with lids, beakers or measuring cups, water, a light source, a dropper or a measuring teaspoon, a rubber stopper or something to seal up your test tubes, foil to make it dark, a drinking straw, and wet paper towels for cleanup. All right, now let's think about our hypothesis together. So in this experiment that we're going to do, we're testing. Let's go back to our objective. Let's look at here what our objective was right there, to observe how plants utilize carbon dioxide and release oxygen during photosynthesis using bromothymol blue, a pH indicator, to detect changes in oxygen or carbon dioxide concentration. Now you read here what happens as carbon dioxide dissolves in water, the pH gets lower. All right, so given all that, what could be a potential hypothesis for this? What do you think will happen to the color of that bromothymol blue solution when photosynthesis happens in the light, in the presence of a light source? Shmuel.
If photosynthesis happens in the presence of a light source, then the concentration of carbon dioxide in the water will decrease.
Good. So one thing that we're going to change is light, right? One of them is going to be in light, one of them is going to be in the dark. That's one hypothesis. We are independent variable. That's one thing we're changing. We're actually changing 2 things in this one. This is not really, we could run these separately. We're just putting them together. So it's not really a controlled experiment because we are changing 2 things. What else are we changing besides light? The plant. We're going to put a plant in one of them and not in the other. All right, so this could be a two-part hypothesis. So our independent variable is either the light or the plant or both. Our dependent variable, what are we going to be looking for? What change are we observing and watching for? What is our dependent variable? Yeah, the color. We're going to be looking for color changes in the bromothymol blue. Exactly. All right, I'm going to read a question here. Marianne's got a question. All right, if pH levels are based on how much hydrogen or hydroxide ions are present, and bromothymol blue is a pH indicator, why does it change color depending on the amount of carbon dioxide present? Good. Why do you think? Take a look here. Let's look at this color chart. That's an excellent question. What happens to the color of bromothymol blue at low pHs? It turns this yellowish green. What happens at high pHs? It's going to be blue. So if it changes color based on carbon dioxide and it becomes more yellow, what do you think that indicates about carbon dioxide? Is it acidic or is it basic? Going to be basic, or I'm sorry, acidic. Carbon dioxide is acidic. When we start with bromothymol blue and we add carbon dioxide, we're going to make it lighter colored. Carbon dioxide is an acid. It's actually a really, really powerful acid. It's a big reason that if you hold your breath, you start feeling bad pretty quickly, because you have sensors all over your body that detect carbon dioxide levels. And as soon as those levels start going up, your body does all kinds of things. Your brain sends messages everywhere trying to get you to take a breath, to blow out that carbon dioxide. Yeah, your breathing, your feeling like dizzy and all that is way more about having too much carbon dioxide than not enough oxygen. You can live longer without oxygen than you can with higher carbon dioxide levels, because carbon dioxide is so acidic, it starts doing damage pretty quickly. So we breathe in order to get that carbon dioxide out. All right, excellent question. All right, now let's read through our procedure. Again, always read. Oh, I'm sorry, Shmuel, I didn't see the hand.
I think the question was that how is carbon dioxide acidic if it doesn't have more hydrogen atoms, it just has carbon dioxide.
So, okay, all right, hold on. Carbon dioxide. What it's going to do, remember when we mix things with water, they react. Sometimes things just dissolve and they don't really undergo a chemical change. Sometimes they do. In this case, the bromothymol blue is going to undergo a chemical change. So bromothymol blue is going to contain some hydrogen and it is going to bond with the oxygen and make hydroxide, or the hydrogen that comes from the bromothymol blue, depending on whether we're talking about a low pH with more hydrogen or a high pH with more oxygen. All right, does that make sense? All right, thank you for clarifying that. Thank you. All right, so let's read our procedure. The first thing we're going to do is put on our safety goggles. Then we're going to prepare the bromothymol blue. We're going to put about 5 ounces of water, that's a little more than half a cup, into a 2-cup measuring cup. The reason is because we're going to be blowing on it. We don't want it splashing everywhere. We're going to add 5 to 10 drops of bromothymol blue. We may need to add more in order to make it blue enough. We'll have to play around with that. We're going to observe the color change. We're going to then gently blow into the bromothymol blue solution using a straw for about 30 seconds. We're going to blow long enough to add carbon dioxide to make that solution a greenish yellow. Then we're going to pour the carbon dioxide rich. We've blown in it now. It's this greenish yellow. We're going to pour that into 3 test tubes or jars. One of them we're going to label no plant. One we're going to label experimental light. And the other we're going to label experimental dark. All right, so hopefully you got a marker and you can in some way, I have mine, I wrote control on that. If you don't have a marker to write on your jar, you can just write it down on a piece of paper and sit it there by it. That will work. All right, and the next thing you're going to do is place a plant in both of the experimental test tubes and seal them with a stopper or lid. You're going to leave the control test tube without a plant and you're going to seal it with a lid. All right. Then you are going to wrap the experimental dark in the foil, and that will block out all light. Without light, the light reaction can't happen. Then you're going to place all 3 of those test tubes under a light source. And then you're going to use the chart I'm going to show you in a minute to look at the initial color of the bromothymol blue and record any color changes at intervals of 15 minutes, 30 minutes, 1 hour, approximately 8 hours, and 24 hours. So for those of you guys who live on the East Coast, I know 8 hours from now, you're probably in bed. So that's fine. Just look at it before you go to bed. For those of you not doing the experiment, I will send you pictures tomorrow of what mine look like at 15 minutes, 30 minutes, 1 hour, 8 hours, and then tomorrow at the end of the day, 4 o'clock, I'll send you the 24-hour one. All right. So now that we've read all of them, we're going to, okay, we're going to do the experiment. All right. First thing we do, put on your safety goggles. Hold on. I have a question here. No, you don't have to do the hypothesis. We did all this together. You don't have to write a lab report on this. This is just for us to do here. Okay, so put on your safety goggles. And I'm going to turn hold on just a second here. I'm going to turn my camera down so you guys can see the experiment as I run it. Get my whiteboard. And you can observe what's happening here. Okay, so I have my 3 test tubes. I have them labeled control, experimental dark. I put an E and a D, and experimental light, an E and an L. I'm going to take off the lids so that they're ready to go. I have my water in my beaker. This is actually a flask. This narrow neck is called a flask. I have my bromothymol blue right here. Oh, Shmuel.
Is 150 milliliters for all 3
Yes.
combined, or
Yep.
each?
No, it's enough for all 3.
Okay.
For all 3 test tubes. Yep. All right. So first thing I'm going to do now is put 150 milliliters of water into my measuring cup. I've already got mine done here. So if you didn't, then go ahead and do that. Put your 150 milliliters. It's a little more than half a cup. So like half a cup and then a little bit more water. Next, we're going to add the bromothymol blue. So mine came with a dropper. If yours didn't come with a dropper, you can just measure about a teaspoon and then pour it into your measuring cup. So I'm going to put mine in and watch the color change of the water. 1, stir it in, nothing happened. You can see that. It's kind of reflecting these blues here, but put another drop. Again, oh, it's got a little slight, very, oh, gone. It's gone again. 3, 4. Now I'm starting to get a little bit of a shade of blue. Can you guys see that? Little blue. So I'm going to do the rest. 5, 6, 7, 8, 9, 10. And I will take my straw that I'm using and I will stir. Now I've got blue water. Okay. To make it a little bit more dramatic, if yours is that blue, that's fine. I'm going to add a little bit more in mine so that you can kind of see the blue. It'll get to the point where you can keep adding and it won't turn any more blue. So taking a look at this now, you know, remembering that chart that we looked at before, let me go back to that. Right here, I'm looking at a pH of about 7.5 in mine. It's about that color. And I'm actually going to stop the share for a second here so that you can see the, hold on. And I can pin this so you can see it bigger. Okay, there we go. That's better. So you can see that. All right, now, blue, I want to add carbon dioxide. I want to make it carbon dioxide rich. And the way I'm going to do that is by gently, so that I'm not splashing all over, I want to gently blow into this bromothymol blue solution for about 30 seconds. Might take more, it might take less, really depends on, you know, the amount of carbon dioxide that's in my breath. So let's begin. You guys can watch as the color change begins. A little bit there. See, it's turning a little bit green. significantly green now. I'm gonna see if I can get it just a little bit more. All right, we've got a yellowish green there now. You can definitely see the color change, right? From what it was before. All right, now our next step is going to be to pour this carbon dioxide rich bromothymol blue into the 3 test tubes. I'm going to pour some in the first one, put approximately the same amount in each of your jars or test tubes. I've got test tubes that are 50 milliliter ones, so I'm going to fill it to about 45. I want to make sure I leave enough room for a plant. Now I just did the control one. So the control, I'm not going to put a plant in. So I screw on the lid. Right there, that's done. I can lay that aside. Now I'm going to put it into the experimental dark and the experimental light. And my next step now is going to be to place a plant in both of these experimental ones. So I have my plant right here. And I'm going to take a piece. I want it to be able to go down into all the way in. So it has to be short enough that it'll fit all the way in. So I put that plant down in, and then I put on a lid. All right, now what I'm going to do is I'm going to fill this the rest of the way up with my water. There we go. Just makes it easier to see to do that. And now I'm going to take my second plant and put that down in my other experimental tube. This will work with a jar. When I lay it down to take a picture of it, if it's not all the way full, you'll just see the air bubbles. So, all right, there we go. So now I have my experimental tubes, experimental light and experimental dark. I have my control tube, and you can see they're all the same. They all have approximately the same color. You can see the plants in these, but the water is the same color. Now I have a piece of foil. I'm going to take the foil and I'm going to wrap the experimental dark tube in foil. Doing that, the ED experimental dark, wrapping it in foil all the way around so no light can get in, is going to prevent the light reaction from happening. Okay, there we go. Now, just to keep things clear, I'm also going to write control. I know it's upside down for you guys. Experimental light and experimental dark. And I will keep these on here as I go through the day of this experiment, the 24 hours that this lasts. And now we're ready. I'll take these, I will put them under a light source. So by light source, what I mean is you can leave them in a bathroom with the light on. If you've got a window, as long as it's daylight, you can leave it under the window, and then you just want to have it in some kind of light all through the night, because remember, light's needed for photosynthesis to happen. All right, hold on 1 second here. Now I'm gonna stop this pin. Here we go, remove pin, and I'm going to share my screen again. There we go. Okay, Shmuel.
But if I do it in jars, my jars are like more than 350 milliliters, so the water will like barely cover it at the bottom, and then how will the carbon dioxide just be still drawn out?
So the more air there is in the top, oh, I'm still not looking at you. Hold on. There we go. The more air there is at the top, the less the carbon dioxide is going to have an effect. So if you've got a big jar, I would mix more of the bromothymol blue and blow into it again, and keep doing it until your jar's at least 3/4 full of the water and bromothymol blue.
Okay.
Yeah, that's a very good point. So like if I had this size jar right here, I would want to fill it until it was at least 3/4, because whatever oxygen is up there is going to dissolve right down into the water.
So just keep them adding in more water and bromothymol blue and blowing until it's.
Yeah. Yep. So you can do the whole thing again. Just do the first 2 steps, 3 steps. make your bromothymol blue, gently blow into it and keep adding that same kind of yellowish green until they're, you know, 3/4 full.
Okay.
Yeah, excellent. All right, so again, so if you got any of this on your skin, use the paper towel to wipe it off. If you got a lot, go wash your hands right now. But if you have just a little bit, use the wet paper towel that I told you to bring. Now, I will tell you guys, we are going to be doing at least 1, maybe 2 more experiments this year with bromothymol blue. So it'd be a good idea to buy some. It's not that expensive if you buy it from one of those companies that takes a little bit of time. Amazon, it was more, but like Home Science Tools is a great place to buy stuff. And having the bromothymol blue will make it a little bit more interesting. So we're going to fill out this chart right now. We're going to fill out what we can. So hold on 1 second. Alexa, set timer for 15 minutes. 15 minutes. All right, we won't be together at 15 minutes. So I will send you tomorrow about the same time as we are now, same time of day. I will send you all my results. So I'll send you pictures of my results here so that you can look at it if you weren't able to do the experiment. All right, so we're going to write in the initial, the color of the control. In the initial, they were all that pale yellow or yellowish green, whatever color you think that is. Yellow, pale yellowish green, pale mint green, something like that. So you're going to write that in there. At 15 minutes, you'll look at my one that will be labeled 15 minutes and you'll write those colors in, or look at your own and write the colors in. That's what you're writing down, is how does the color change? All right. In the last few minutes that we have here, while we're waiting for 24 hours to go by, I want to ask some discussion questions. So what color was the original solution of bromothymol blue in water originally? When I first mixed the BTB in with the water. Yeah, it was blue. It was that nice, pretty blue color. All right. Why did I put goggles on? I already talked about that. The reason is because it could irritate my skin or my eyes if I got on it. So you want to make sure you don't do that. All right. Why did I make a control? One that had carbon dioxide, but no plant. Why did I keep one with no plant? Yep, to make sure the experiment goes well, but how, in what way? To determine if a change really happened. Yeah, to see what changed and what didn't. Yes, to see what I'm trying to see, does the plant make a difference? Excellent. So why did that color change to greenish yellow when I blew into it? That's what bromothymol. Oh, go ahead, Zoe.
Because you added carbon dioxide?
I did. I added carbon dioxide. I made it more acidic with my carbon dioxide. So that turned it into that yellowish color when I blew the carbon dioxide in. Okay, why am I using a light source? Why is that one of the things I have to do in this experiment? Yeah, so that photosynthesis can occur. Remember, the first stage of photosynthesis requires light. That's why we call it the light reaction. If I just put these all in the dark, nothing would happen. They would all stay the same because photosynthesis would not be happening at all. All right. What comes off of a plant? Sorry, what comes off of a plant during photosynthesis? What does a plant release? Oxygen, exactly. Oxygen. What color is bromothymol blue when there is oxygen? And not so much carbon dioxide. It's going to be blue. That original color, when I originally mixed it in, that was because my water had oxygen in it. So it turned blue. It turned that bromothymol blue, the bromothymol blue turned the water blue. What color do you expect to see in this control test tube at the end? I'm not seeing the correct answer. Basically, what color it is not going to change. Nothing should change in this one. It should stay exactly the same. Now, if it changes to a blue color, that probably means I don't have the lid on right, or it's not a tight seal and oxygen's getting in. As long as I've got a good seal, nothing should change. What color do you expect to see in the tube we wrapped in foil? Once again, it should be that greenish yellow. Nothing should change because I'm not allowing photosynthesis to happen. I'm preventing it with the foil. And then finally, what color do you expect to see in the tube that contains the plant and was exposed to the light source? Yeah. Yeah, that's what we're hoping. That's what we're hoping for. We're hoping that the plant gives off oxygen, that the plant takes in carbon dioxide, and it returns it to that blue color that I had before. Now, you're going to look at your experiment in 15 minutes, in 30 minutes, in an hour, and then again in right before you go to bed, and then again in 24 hours. Be sure to complete your chart and fill out those analysis questions at the end. You don't have to turn your lab sheet in, but I want you to bring it to class next week because we're going to talk about our results. So I expect everybody to have completed their charts and did the analysis questions, whether you did the experiment or whether you just observed. Okay? Okay, Shmuel will not turn yellow. So you're blowing into it and putting carbon dioxide in and it's not paling up. Can you hold it up and let me see it? Okay, I would say keep go. Did the first time, when you did it the first time, did it have? No.
This is the first time.
What?
This is the first time.
Okay. I just, here's something. All right, I'm gonna hold on for a minute after class, Shmuel. I'm gonna give you a hint, but I don't want everybody else to hear it because it'll come up later. Okay, so you just hold on. Everyone else, you are free to go. I went a couple minutes over. I hope that's okay. All right, make sure you fill out and bring back your analysis questions and your chart next week. Have a great week, everybody.
Thank you.
Bye. Thanks for coming. Bye. Okay, let me stop the recording here. Hold on a second. More. Stop. Okay.